Hello, thank you for joining us today. I'm Tracy Smith, the Collaboration, Education, and Outreach Coordinator for the Center for Integrated Cellular Analysis here in New York City. In this seminar series, we highlight new papers and methods from our center that we believe to be of interest to the broader community. Today, we focus on the reconstruction of forebrain evolution in tetrapods. This work uses single-cell and spatial atlases of the salamander brain to explore the evolutionary origins of the vertebrate neocortex. We'll have time at the end of the talk for questions, and we encourage you to either raise your hand and I'll ask you to unmute yourself, or you can type your question into the chat and I'll read it aloud to the speaker. Our guest today is Jamie Woych. Jamie received her BS in biochemistry from SUNY New Paltz and has been the lab manager of the Tosca's lab at Columbia University for the past 3 years. Throughout this time, her research has focused on the spatial distribution of glutamatergic cell types within the salamander forebrain. She's specifically interested in how evolution has shaped the innovations and complexity of present-day nervous systems. Jamie, thank you so much for being with us today. I will hand it over to you.
Screen sharing. All right, so thank you for the opportunity to share this work. This is the first preprint coming out of the Tosca's lab. And thank you to everyone who came here today and joined this meeting. So first and foremost, I want to acknowledge the team that brought this work together. This is Alonzo, Eliza, Astrid, and Eli. And this was just a tremendous group effort, and I'm incredibly grateful to be a part of this team and a part of this work that we put out together. So jumping into the paper, I'm just going to step back and talk a bit about just the overarching goal of our work in the lab and of neuroscience in general. So what we want to understand is just generally how the brain works and how it functions. And studying brain evolution is a really integral part of this work. So in order to understand the anatomical organization and function of the mammalian brain, we first have to look at the general principles and basic building blocks of brain function in a simple brain. And these general features and principles arose across the course of evolution due to constraints and favored pathways that acted throughout evolution. So when you look at this phylogenetic tree of various vertebrate brains, despite 500 million years of evolution along this tree, you see an amazing amount of conservation of architecture. So if you look here at these regions, in orange, we have the telencephalon, in green, the midbrain, and in purple, the hindbrain of all of these very different animals. And you see this conservation of architecture despite very species-specific differences in cell types and circuits. Today, we're going to be focusing on this branch here, comprised of all of the tetrapods. And if you take a slice coronally of a brain here, you would see something like this, here showing a coronal section of the salamander, turtle, and mouse brain. And what's highlighted in blue here is the dorsal telencephalon, also called the pallium. And within the pallium, 4 compartments have been classified and identified within all vertebrates. And these are based on their developmental origin. So you can identify a medial pallium, a dorsal pallium, a lateral pallium, and a ventral pallium. And zooming in on the dorsal pallium of these species, which has had a great amount of diversification throughout the evolutionary history of vertebrates, you can see in mammals, we have a 6-layered neocortex that is involved in advanced cognition in mammals. And the neocortex evolved from an expansion and diversification of an ancestral 3-layered cortex, which still exists in reptiles. Another feature of the pallium in reptiles and also birds is the DVR. So the DVR is also involved in advanced cognition within these animals and shares some connectivity patterns with the neocortex. But when you look at development, it develops from an embryonic ventrolateral pallium. The embryonic ventrolateral pallium in mouse, however, gives rise to areas such as the claustrum and parts of the amygdala. It's unclear whether homologs of the DVR exist in mammals, and homologs existing in salamanders are not really explored yet. So that brings us to our model organism, Pleurodeles waltl, also called the Spanish ribbed newts. And in comparison to reptiles and mammals, these newts have very simple and small brains. So here I'm showing a picture of the salamander brain in comparison to a mouse brain, and it's, as you can see, it's much smaller. And the size and simplicity of this brain makes it relatively easy to do molecular analyses of the entire telencephalon. So again, taking a coronal section here, when you zoom in on the dorsal pallia of the salamander, you don't see any obvious layering architecture, and the pallium is devoid of any large nuclei that you would see in other vertebrates. This makes it a very useful model for making comparisons across species. Sharing a common ancestor with amniotes about 350 million years ago. This node represents the transition from water to land, and environmental changes at this point likely led to innovation and adaptation within the vertebrate nervous system. Origins of pallial regions in mammals and reptiles, such as the neocortex and the DVR, likely can be traced back to this common ancestor and cell types that existed then, or are novel features that arose at nodes throughout evolution. So historically, cell types were classified based on comparisons of morphology and physiology. However, cell types are genetically determined by core regulatory complexes and effector genes. So cell types can only be classified as homologous when they're inherited from a common precursor, but cell types can arrive at the same phenotype through cell type convergence. So knowing that cell types are homologous gives us information about basic features that are conserved over these very wide evolutionary splits. And that lets us know what are these basic principles of brain function. However, if cell types have arrived at the same phenotype multiple times throughout the course of evolution, that gives us information about constraints and favored pathways that act on development and anatomical organization. Alternatively, cell types that are novel gives us information about what makes different brains unique and what allows for more complex function. So considering how long we've been trying to understand brain evolution, single-cell transcriptomics is a relatively new approach to comparative neuroscience. And it allows us to look at comparisons of cell types, not based on morphology and physiology, but based on genetic programs and molecular expression. And we can look at these deep splits and also compare developmental gene programs as well as connective similarities within these brain regions. And we can ask questions utilizing our cell type atlas, such as how do developmental programs diverge to produce this diversity that's seen in the vertebrate brain? Do these pallial subdivisions share molecular signatures across species? And do neuron types with similar transcriptomes share similar circuit motifs across species? So first we had to build our cell type atlas. So here we're showing a portion of our data set, including only the glutamatergic neurons of the telencephalon. And we can use this data set to find marker genes of the different pallial regions within the brain, such as LHX9, ETV1, LHX2, and SOC6. We can then map the expression of these marker genes spatially in the tissue using histological techniques such as in situ hybridization, shown here. And we can map these boundaries of the pallium based not on what little morphological landmarks exist within the tissue, but on differential gene expression we've seen throughout our data set. And something that we found that was very interesting is the gene expression profile of this marker gene, NTS. And you see that it has high levels of expression in the 3 different clusters in the medial, dorsal, and ventral pallium. And when you look at the expression pattern of this gene, you can see here in the medial, dorsal, and ventral pallium. And for example, when you zoom in the dorsal pallium and compare the expression of NTS to other markers of the dorsal pallium, you see that its expression is much further away from the ventricle than, for example, ETV1, which is another marker of the dorsal pallium. So in addition to this medial-lateral axis of expression profiling, we also see a radial axis of expression. And this is showing cell type layering in the telencephalon that has not previously been classified in salamanders or amphibians. So another thing we were able to do is leverage the size of the salamander brain. And we used HCR in situ hybridization, iDISCO clearing, and light sheet imaging in order to visualize these gene expression patterns in whole tissue volumes. So what's really cool about this is we can then compare these very distinct regions, here showing this dense band of the lateral pallium extending from the olfactory bulb to the most posterior region of the telencephalon, to ETV1, which is expressed in the dorsal and medial pallium, to PENK, which is expressed only in a portion of the medial pallium and also the striatum. And it's very helpful to be able to visualize this gene expression and slice the brain in different stacks in order to look at the distribution of each of these regions. So because cell types are the products of development, we also wanted to look to see if these programs diverge to how these developmental programs diverge to produce diversity that's seen within the vertebrate brain, especially considering the amount of diversity we've seen in our cell type atlas. So we sampled 4 different stages throughout development for single cell RNA sequencing, here showing stage 36, 41, and 50 schematics, but we also sampled stage 46. And then just a simple immunostain showing the radioglia and the differentiated neurons marked. So we then mapped our adult data set onto our developmental data set using Seurat label transfer. And this UMAP is showing that our larval data sets contain differentiating neurons that belong to each of these broad subdivisions of the adult subpallium and also the pallium. And again, zooming in on the pallium, we then inferred developmental trajectories of these differentiating neurons using slingshot trajectory analysis. And here you can see that these 4 subdivisions of the pallium, medial, dorsal, lateral, and ventral, all have very distinct developmental origins. So this is kind of a molecular confirmation that each of these different regions, which have very distinct molecular signatures, are also coming from different differentiating cells. So being interested in also these genetic programs, we looked closer at the gene expression profiles of the dorsal and ventral trajectories. So we reordered these cells based on pseudotime and then looked at the genes upregulated in each of these trajectories. So here showing genes upregulated in the dorsal trajectory and genes upregulated in the ventral trajectory. And you can see in comparison, genes that are upregulated in the dorsal trajectory are not upregulated in the ventral trajectory and vice versa. Here we have annotated all the transcription factors that are in each of these heat maps, with PBX3 and SOC6 being transcription factors that are also upregulated in the development of mouse ventral pallium. So not only do these regions have distinct developmental origins, but they also have distinct developmental gene regulatory programs that are likely relying on these evolutionarily conserved transcription factors. So considering these developmental origins, we also wanted to see if the pallial subdivisions between species shared molecular signatures. So we first integrated the telencephali of salamander and 2 reptiles, lizard and turtle. And these UMAPs here are color-coded by the black cells are cells that here are in the salamander, here in the lizard, and here are in the turtle. And when you color code the pallial portion of this UMAP by pallial region, it looks like this. So with this here, you can see that these compartments of the reptilian pallium, medial, dorsal, lateral, and ventral, correspond to their counterpart in salamanders. And this is based on molecular similarity that could be due to homology or convergent evolution. But I want to point out notably the similarity between the ventral pallium and the anterior portion of the DVR in reptiles. Which is interesting. It'll come up later with our connectivity data. So we also used different integrative methods to verify our results. And utilizing SAMMAP, Harmony, and SciBi, we had very similar results for each of these methods. And this is just confirming that these molecular similarities are robust and not just due to artifacts of the integration method that we chose. We then did a similar integration including mouse, but only including the dorsal medial pallium of each of the species as well as interneurons. And this is just due to the fact that the mouse data set does not have complete telencephalic data. So when you look at the UMAP, it looks like this, and each of these dots represents a cluster that's color-coded by the proportion of cells that are coming from each animal, and the size of the dot represents how many cells are in each cluster. So this is just another way to visualize our integrated data set is as a tree that's hierarchically clustered. So on the left side, we have the GABAergic cells, and you can see that as they're colored by proportion contributed from each species, we have a lot of gray branches on this left side, indicating that they have cells that are coming in equal proportions from each of these species. And this implies a high level of conservation of these cell types. Whereas on the right side, you see many of the branch points are primarily blue or completely blue, meaning that the cells in these clusters are coming almost entirely from mouse, which is pointing to a lot of novelty on the side of the glutamatergic cells contributed in this integration from mouse. So when you zoom in on these glutamatergic neurons, we have here below these shadings that represent the proportion of cells that are contributed from mouse that are grouped by projection neuron type. And when you look at cells that are coming from each of these projection neurons, you see that they coincide with these splits and branches, these large splits within the tree. And then when we reorder these clusters based on mouse cortical region labeled here, you can see that the salamander dorsal pallium, as well as most of the turtle dorsal cortex, aligns with regions of the mouse and co-clusters with clusters that contain mouse hippocampus, mouse subiculum, and entorhinal cortex. In addition to this, you see that a portion of the reptile dorsal cortex aligns with these L4 thalamo-recipient IT neurons. So when you look back at the UMAP, we're going to zoom in on these clusters 10 and 12. And based on the color of them, you can see that they have contributions fairly equally between the four species. And here we've recolored them based on their species of origin and annotated where these cells came from in the original data set. So in cluster 10, we have the reptile dorsomedial cortex co-clustering with salamander medial pallium and mouse entorhinal. Whereas in cluster 12, we have the dorsal cortex of reptile aligning also with the dorsal pallium of salamander and the mouse subiculum. So the three main takeaways we can have from this four species integration is that the salamander dorsal pallium lacks any molecular characteristics of the mammalian neocortex. However, part of the reptile dorsal cortex co-clusters with these L4 thalamo-recipient IT neurons of mouse, while most of the cell types that are in the mammalian neocortex have arisen due to innovations in the mammalian brain and are completely novel to these comparisons. So after making these comparisons, we also wanted to look at circuit motifs and whether neuron types with similar transcriptomes also shared similar circuit motifs across species. So we did this by injecting a retrograde tracer into various regions of the amphibian pallium. And here you can see that we injected this retrograde tracer into the ventral pallium and then saw retrogradely labeled cells in the thalamus, in the lateral pallium, and in the accessory olfactory bulb. And then when we compare this connectivity pattern to the region in the reptile that has similar molecular features, we see that they share connectivity patterns as well. So both of these regions project to the hypothalamus and receive afferents from the thalamus, the dorsal pallium, and the lateral pallium. When looking at other regions of the pallium, you see the lateral and dorsal pallium projecting to the medial pallium, as well as the olfactory bulb projecting to the lateral pallium. And when you compare this connectivity to the regions that are molecularly similar in mouse, you see this conserved olfactory entorhinal hippocampal circuit that's found in mammals. And this is outlined here in the schematic to the right. So we can take this data and look at it all together in combination, looking at the development and the connectivity and the molecular similarities between all of these branch points in the evolutionary tree, and make this hypothesis on how cortical neuron types in vertebrates have evolved over time. And you see these similarities between the dorsal pallium of amphibian to the dorsal cortex of reptiles and entorhinal subiculum in mammals. And you also see connectivity and molecular similarities between ventral pallium and this anterior portion of the DVR. But you see that there are also innovations in the forebrain where amniotes share these dorsal cortex neurons between reptiles and mammals. But for the most part, we see that innovations in the neocortex are what gave rise to novelty that's seen within mammals. So to wrap this up, in order to understand these conservation and innovations within the vertebrate forebrain, amphibians are a really critical comparison point to understand brain evolution. And looking at our data set, we see a much greater amount of diversity than can be anticipated from looking at the general architecture of the amphibian brain. The dorsal and ventral pallium have very distinct developmental origins and gene programming that could have connotations for conservation of genetic programming throughout all vertebrates. And the ventral pallium of amphibians shares molecular and also connectivity similarities to the reptilian DVR. Much of the neocortex arose from novel innovation of cell types in mammals, and we can continue to leverage this relative simplicity of the amphibian brain to look at how these conserved circuit motifs and cell types contribute to behavior and function of vertebrates today. So that concludes my talk for today. I just want to again thank everyone who's in the Tosques lab, and especially our wonderful PI, Maria Antonieta Tosques, and all of our co-authors who were involved in this work. And we're ready to open up for questions at this point.
Great, well, thank you. That's incredible work, Jamie. Really beautiful images that you shared with us. We'll move on to our Q&A. So if anyone would like to ask a question, you can raise your hand or type your question into the chat box. And we have a question here right away. Do you find evidence where the molecular homology has diverged, say the gene marker switches its cell type between amphibians and mammals?
So,
and Socket, if you're able to clarify a little bit, feel free to do so.
Yeah, sure. Yeah, so my question essentially was, so basically your analysis is based on using sort of homologous genes across all the species and looking at them all through at once. But if you were to focus on gene markers individually in different species, you find evidence that a particular gene, which is actually a marker of, let's say, a particular subcluster of the glutamatergic cell type in salamanders, is actually marking a different cell population in mammals.
Okay, I'm understanding your question. So in order for cell types to be homologous, they need to be inherited from a common precursor. So to have molecular markers that are being expressed in different cell types would point more to convergent evolution, right? So I guess we see these Rohr beta cells that I didn't really talk about expression of Rohr beta, but these are the dorsal cortex cells that are shared between reptiles and mammals. And in amphibians, for example, Rohr beta is not expressed in the dorsal cortex or in the dorsal pallium at all, but instead it's expressed in the lateral pallium. So does that?
Yeah, that does.
So these are like deeply conserved markers that are well known in reptiles and mammals, but when we looked at the expression patterns in salamanders, it's very different.
Right, and just to get a ballpark, the numbers of such switches are small, is what my guess is. Is that right?
The number of
so the number of such genes that you see, which are sort of differential markers in some sense. Do you have a guesstimate of?
I don't have a number.
Okay. Yeah, that's fine. Thanks.
Great, thank you. We have another question here in the chat. Is it known if polyploidy in salamanders or other animals is related to increased neuron diversity? So example, do you think polyploidy in salamanders might be affecting homology between regions because it's related to increased diversity?
So I think you can argue that there's less diversity in neuron types in salamanders. You see, these